Welcome to a few minutes of RNA expertise with Lexagen. Today we will present CORAL. CORAL stands for Cover All Bases and is Lexagen's highly versatile, fast, and robust RNA-seq kit for samples from all sources, including low-input RNA, degraded and FFPE samples. CORAL is a one-of-a-kind fragmentation-free library prep. It uses Lexagen's proprietary displacement stop technology to generate defined insert sizes, avoiding fragmentation bias. Random primers are elongated until the next primer is reached and the reaction is efficiently stopped. Unique molecular identifiers, or UMIs, are introduced in the second step. UMIs allow the removal of PCR duplicates for accurate quantification of transcripts and genes. UMIs are conveniently located at the beginning of read 1 and are thus accessible using cost-efficient single-read sequencing mode. CORAL libraries can be multiplexed using 384 unique dual indices for maximized sequencing output. Plug-and-play, non-expert data analysis solutions for CORAL are available online, allowing everyone to analyze the data from their sequencing project. CORAL is available as mRNA-seq with poly-A selection and total RNA-seq with ribodepletion. Are you curious to try CORAL? Contact sales@lexagen.com for a quote, or consult with us at support@lexagen.com about your project.